Good morning. My name is Jean-François Mounet. I'm the CEO and the co-founder of GENFIT. First of all, I want to thank you in the room for attending this meeting. And I want also to welcome the audience via the WebEx. And I know that a lot of European investors, individual and institutional investors are waiting for this WebEx. So we had, during the last JP Morgan week, the idea to organize this R&D event because a lot of investors that we have met or analysts, they raise many questions about the strategy of GENFIT in the future years, and they also ask about what else that Nash and your phase 3 Nash, do you have other programs? So we have decided to present all the pipeline of GENFIT, and sometimes I know that you will probably discover some things. I want to say also that we have a press release which has been published a few minutes ago, and where we announced some important things that we will present during this meeting. So I want also to introduce my colleagues. Robert Valzac is the Vice President of Research. Sophie Ménien, our CMO, now in Cambridge office. And Dinem, our CSO. But I want to particularly thank the Dr. Manal Abdelmalek. She's an hepatologist, a liver transplant specialist. She's associate professor at the Duke University Health System. And probably, as you know, she's a very renowned as a lead investigator in many Nash trials, and she belongs to the Nash CRN. So we will go through that disclaimer very quickly. And then to speak a little bit about our strategy. Our strategy is to have an evolution of the company from a biotech R&D company to a biopharma dedicated to specialty pharma, and we want that to be, I would say, very coherent. We want something to be the crossroad between GI, metabolic, and autoimmune inflammatory diseases. So as you can see, of course, we're in Nash. We have also some program in fibrosis, cirrhosis, cholestatic diseases. We have announced, and we will announce and give you more details about the fact that we are launching a trial in PBC. We're working on different hepatitis, autoimmune hepatitis, and we are exploring also some additional therapeutic area. Gastroparesis is just an example of things that we are interested in. In a snapshot, I would want to comment this the pipeline, and you will get, of course, more details during this meeting. First of all, the NASH program. As you know, it's a most advanced program. It's a drug development program in phase 3 now. We have enrolled the first patient last month, and we want to address this huge unmet need and probably what will be a blockbuster market. In parallel, as you know, we have always been pay a lot of attention to the fact that you cannot imagine to access the market and, of course, to treat the patient if you cannot diagnose them. And we could not envision to do that at a large scale with the biopsies, too invasive. So it's why we are working for years now in a biomarker program. It's a key for us to unlock the NASH market, and we will tell you more about that. And we will also give you the identification of the different biomarkers of the algorithm. The PBC. The PBC is a new leading program in GENFIT with a potential second indication for elafibranor. As you know, it's an orphan disease. The question is always, it's still an unmet need, and we think it will be an unmet need for many years, because even if there are some drugs to treat the patients, probably you need different drugs to address the patients with different concerns and additional cardiovascular concern. For example, could be some example of what we can bring as an added value on other drugs. We have programs in NASH, and Sophie will give you more details about the NASH pediatric programs that we will launch this year, and about some program we envision in cirrhosis. And then we'll give you our approach about combo therapy. We believe strongly that the combination therapy will reinforce the competitive advantage of elafibranor. We consider that by combining complementary mechanism of action, we can address the drug to more people, to more patients. We have also some research program targeting end-stage liver disease with 2 liver fibrosis cirrhosis programs, and one of these programs will be launched in phase 2 also this year. We will have more information about that. ROR-gamma T is a very important target we are working for years now. It's a very hot subject and dedicated to autoimmune inflammatory disease. We are thinking about an indication which is autoimmune hepatitis for this drug. And finally, just to say that we are working very hard with our team about exploring in-licensing opportunities to continue to develop this strategy. I mean, a strategy of a coherent pipeline, very diversified, but coherent in the field of gastroenterology and metabolic disorders. So now we are going to go through the different lead programs, and I would like Manal Abdelmalek to present the next slides, please. Just technical few words, I'm sorry for that. Just to say that we will provide a slide deck more detailed on the website of the company during the day. And that you have some papers in front of you. If you want to write some questions during the presentation, we will address some of these questions during the Q&A session. Okay, thank you for that. Sorry.
Thank you, Jean-Francois. I thank GENFIT for the opportunity to be here with you guys today in New York. Just a little bit of background about myself as it pertains to my interest in NASH as a space. I've been in this area of academic interest since 1995 when we reported the first case of NASH progressing to cirrhosis with clearance of fat. And my career since then, for the past 2 decades, has been academically invested in this space from a clinical research perspective, patient-oriented research, and cross-collaborations with many people to try and define mechanisms for the disease acquisition and progression. So it's wonderful for me to get to an opportunity in my career where I'm seeing therapeutics in play. So clearly, NASH is the major underlying cause of progressive fibrosis currently in the Western world, and it results in a process of necroinflammation, ultimately leading to hepatic fibrosis progression, and in a minority of patients, advanced liver disease with cirrhosis and complications thereof, needing liver transplantation, or the risk of developing hepatocellular carcinoma. The estimated prevalence of NASH in developing countries right now is approximately 30%, and for NAFLD, benign steatosis, as high as 50%, if not higher. So a huge problem. There are currently no FDA therapies. It's an incredibly high priority for our regulatory authorities in the FDA and EMA right now. NASH is the leading cause of liver disease in obesity, and obesity estimates anywhere from 66% to as high in some cohort studies as high as 90% of obese patients, and in patients predominantly with multiple complications of metabolic syndrome and diabetes. Those that are over the age of 50 in the US have a higher propensity for having advanced fibrosis or cirrhosis. And this depiction here suggests that patients who develop fat in the liver, approximately again 25 to 30% of patients, higher prevalence in diabetics and obese patients, can go on to develop NASH. And NASH currently in the US, based on non-invasive studies, surveillance studies, and Haynes data, is estimated to occur in about 12 to 20% of the cohort. Those that go on to develop NASH will develop inflammation in the liver and over many years progress through fibrosis stages, ultimately to cirrhosis or F4 stage fibrosis, and then have an increased risk of liver-related morbidity and mortality. However, stepping outside the liver domain, patients with NASH are at increased all-cause mortality and predominantly cardiovascular-related morbidity and mortality. Not to mention patients with benign simple steatosis are at increased risk for the incidence, heightened risk for incidence of new onset diabetes mellitus. So NASH is a risk factor for liver failure, cardiometabolic problems, and ultimately increased risk for death. The concern here is this is completely a silent progressive disease, and silent until very late stage signs and symptoms of advanced liver disease occur. The concern is of patients with NASH, the leading cause for morbidity and mortality is cardiovascular disease, followed by non-liver related cancers, all-cause cancers, and ultimately progression to cirrhosis and complications thereof. This is data that came from a former colleague of mine, Paul Angulo, when he was at the Mayo Clinic and evaluated the Olmsted County database. And ultimately what we're seeing over the past, not even a little over a decade, the cause for need for liver transplant for NASH is incrementally rising compared to all other forms of chronic liver disease. And what I can anticipate we would see is the need for transplantation for hepatitis C in the upcoming years will actually decline in the era of new therapies. So GENFIT scientifically addresses the disease through its global approach, providing features of both histologic improvement in the liver and cardiometabolic protection. So addressing two lead cause of potential morbidity and mortality in this cohort. As many of you know, the phase 2 study included 9 countries, 56 centers. We at Duke University were also part of this pivotal study. 3 treatment arms, placebo, and 2 doses of elafibranor. 274 patients randomized, and at the end of the treatment period, the gold standard, which is liver biopsy, was utilized to assess treatment response, and with the definition of NASH resolution without worsening fibrosis. Now, the protocol definitions for NASH resolution without worsening of fibrosis was a score of 0 on at least one of the 3 components of NASH. And the 3 components of NASH on histology are steatosis, necroinflammation, and ballooned hepatocytes. And the progression of bridging fibrosis from stage 0 to 1 and 2 to cirrhosis or advanced hepatic fibrosis. After the study was completed and after the data was analyzed, and as the field was moving forward, we began to recognize that really NASH and the definition of NASH should be redefined. It should be redefined because steatosis in and of itself can flux in and out of the liver and is not the key histologic feature to account for disease progression. So the definition of NASH was redefined more stringently to include the 2 features of necroinflammation that increase risk for fibrosis: ballooning, ballooned hepatocytes, and necroinflammation. So with this new definition, the score of 0 for ballooning had to be achieved, and a score of 0 or 1 out of a 3-point scale for inflammation had to be achieved, with no objective score for steatosis. So in fact, the working definition for resolution of NASH became more stringent and more clinically pertinent, with any progression of fibrosis greater than 1 point being a worsening. The phase 2 study for GENFIT scientifically addresses the unmet need, and the paper was published in Gastroenterology. I was fortunate enough to be a co-author on this paper, and it ultimately concluded that the resolution of NASH without fibrosis or worsening was most effective in the 120 milligram treatment arm, and the results were confirmed with an intention to treat population as well as subgroup analyses for moderate to severe NASH, thus the FDA's recommendation to redefine the NASH resolution endpoint for future studies. And in addition, the publication confirmed that elafibranor significantly improves the cardiometabolic profile and is safe and well tolerated in patients with NASH. So of the patients that were in this study, 274, 12% on placebo. This is a endpoint versus elfibranor, mildly significant, p-value less than 0.05. Of those patients who were reanalyzed with a NAS score greater than 4, again, you can see marked improvement in the elfibranor arm. And when we take a look at the NAS score with any stage of fibrosis, selecting out a more advanced treatment group, the improvement of elfibranor was even more pronounced when compared to placebo. And this is really ultimately what matters. What really matters from a liver-related morbidity and mortality standpoint is what happens with fibrosis. And as we can see here, elfibranor compared to baseline improved fibrosis in those that responded to treatment compared to those that didn't. And when we take a look at other surrogate measures, granted we do not have an ideal biomarker, but if we look at validated surrogate measures to define what's happening in fibrosis, we take a look at the fiber test, Stiato test, or the NAFLD fibrosis score, you could see again a decrease in the surrogate markers for liver injury, and likewise, liver injury is defined by ALT liver biochemistry. Did not affect AST significantly, but ALT is the predominant cytosolic enzyme that comes from hepatocytes. An improvement in GGT and alkaline phosphatase, and likewise other markers of necroinflammation, fibrinogen, haptoglobin, and even C-reactive protein. So it is imperative that any drug that be developed for NASH be at least neutral from treating physician. I treat many thousands of patients with NASH. Most of my patients don't die of their liver disease. They die of their cardiovascular disease. And heaven forbid I put a patient on a medication that will potentially increase their cardiovascular risk. I don't need to be treating their liver disease only to have them subsequently die of a heart attack. So it's imperative that any therapy for NASH really improve and advance their total clinical outcomes, both from a liver-related standpoint and a cardiovascular-related standpoint. And as we could see, the elfibranor arm markedly improved triglycerides, total cholesterol, non-HDL cholesterol, LDL cholesterol, and actually even caused an improvement in HDL cholesterol, suggesting that it may have very favorable cardiometabolic profile. So, GENFIT differentiation currently lies as its first-line potential global approach with efficacy both in liver histology and in cardiometabolic profiles. It's well tolerated with no safety signal. Keep in mind that the unmet need currently is that NASH is a chronic, silent, and potentially progressive, life-threatening disease, and that adherence to treatment is not going to be a short-term therapy. It's going to be probably a very long-term, potential lifelong therapy for the metabolic milieu, and that the drug profile for GENFIT, its phase 2 takeaways, that elfibranor has histologic efficacy, good tolerability, good safety, and not only liver-related improvements, but cardioprotective properties. And that elfibranor has a high potential for first-line treatment in NASH. Thank you.
Okay, good morning, everyone. So going from the phase 2, thank you, Manal, into the phase 3. So just a reminder on the phase 3 design, so the Resolve It trial. We have a first period of the trial, double-blind period, that will be over a 72-week treatment period, double-blind. We have 120 milligram versus placebo. And this first part of the study will use the endpoint of NASH resolution without the worsening of fibrosis. So importantly, the definition used will be the one that Manal explained that came from the academic experts and the FDA, EMEA, on the definition of resolution, meaning we do have to have the stringent definition of resolution through ballooning has to disappear, come down to 0, and inflammation to 0 or 1. So this is the endpoint that is accepted and relevant as a surrogate endpoint to the prevention of progression to cirrhosis, which will be the long-term endpoint. So for this study, we have 1,000 patients for this primary endpoint for the surrogate approval. This will serve for the NDA approval under Subpart H and also conditional approval in Europe. So this has been, of course, validated and in alignment between the EMA and the FDA on the use of this surrogate for approval of the drug on the market. But the study will continue with a long-term extension period. So this is still a double-blind treatment throughout the whole long-term extension phase of the study between 120 milligram and placebo. And the aim is to go to long-term outcomes and events. And these are defined as a composite endpoint of progression to cirrhosis, but also liver-related outcomes. So that means all the decompensation events that are linked to cirrhosis. And importantly, we also have total mortality. And as our expert also explains, in the total mortality, we're expecting mortality from cardiovascular events to occur in these NASH patients. And so this is globally the full design, and a total of 2,000 patients will be enrolled to have the answer of the whole global study. But again, the first 1,000 will be the ones used after 72 weeks of treatment for the submission of authorization to market. This is to show you the timelines, the perspectives with the study. So as you know, we announced recently the first patient recruited into the study. So we have that here. We count the 72 weeks of treatment. The last patient for those 1,000 patients needed for Subpart H approval will be recruited in 1 year. So our estimation today is the 1 year recruitment period for the first 1,000 patients, and then you have to wait 72 weeks of treatment for the readout of the results. So then, of course, this follows with the NDA submission and then approval. This is important that because we have the fast track designation, we are using a rolling review, meaning it's a shorter term for regulatory approval. So we expect this by around mid-2019 for NDA approval due to the fast track, and we will have the other parts of the file and dossier already submitted and reviewed before the final results of the phase 3. Of course, there are other aspects within the phase 3. This is just, the central phase 3 design, but we also have everything around the full, trial development plan up to market of the drug that includes pricing, reimbursement, and market access. And this is going, starting alongside in parallel with the phase 3 clinical trial and developments. And we also, and then Nadine will present more into detail, the biomarker validation and qualification program around NASH, not only with elafibranor, but globally in the NASH field. That's already started, of course, and that will be pursued and continued to be in parallel with the phase 3 study. We're also having a lot of actions in the disease awareness in the NASH field as a whole, and of course the partnership discussions, and Jean-François will talk a little bit more, but all these things are ongoing and, really make the full program around the phase 3 trial. And you have here just the timelines for the estimated market entry. And I will go into more detail a little bit about other NASH studies apart from this phase 3 trial. But first, Dean, I'll come back to the other trials after.
Thank you, Sophie. So what I want to do in the next few minutes is just to talk a bit about our biomarker program in the field of NASH. As many of you may already know, we spoke about this in the past. GENFIT, when we think about NASH, we like to think about a company who's really considering the global clinical management of NASH patients. Okay, as we just heard in our phase 2b trial, what we clearly show with elafibranor is that we not only look at the liver histology, where the compound clearly resolved NASH without worsening of fibrosis, but the other aspect, which is very important, is the cardiometabolic aspect, and there we show efficacy where the compound is able to improve the cardiovascular risk. So, this is really, from a therapeutic standpoint, part of the global clinical management of NASH patients. Now, in the same theme, thinking about NASH patients and the global management of NASH patients, we have to think about biomarkers. We know today that the gold standard to diagnose patients and a confirmed diagnosis of patients as having NASH is the biopsy. Okay, and we all know that that is an imperfect gold standard. All the major stakeholders in the field know this, the regulatory agencies, experts in the field like Professor Abdelmalek, also, they're working on biomarkers as well. So this is something we had built into our phase 2B program. Okay, throughout our phase 2B program, when the patients came in for their clinical follow-ups every 2 months, we had collected blood samples, okay, and stored them away optimally really for biomarker discovery. So subsequent, at the end of our trial, we've taken out those samples, and we really looked into that. So we all know, why do we want non-invasive biomarkers? I think there are different reasons for that. I mean, beyond the fact that the biopsy is inconvenient, there's discomfort for the patients and so on. Another important aspect is that we all realize for the Nash market to reach its maximal potential, it's going to be important to have these non-invasive biomarkers to be able to identify these patients for treatment. But coming back to patient management, I think this is also very important for the patients. As we heard from Dr. Abdelmalek, that Nash is a silent disease, okay, throughout a big part of the process until it's pretty late stage, okay? And this is why what you want to do is get in there, identify those patients who need to be treated before they progress into later stage disease. And this is why we need a non-invasive biomarker. And another aspect, of course, when you think about the economics of Nash, up there we see the numbers here, the cost of performing a liver transplant, okay? And this has really escalated in terms of cost over the last few years. And really the idea is to identify these patients early, treat them, avoid them from moving on to later stage disease requiring a transplantation. So with that in mind, the question we asked with our program was really, we want a non-invasive biomarker to be able to identify patients for treatment. So patients for treatment today, if you think about the phase 3 program, okay, these are Nash patients with a NAS score of 4 or more with F2, F3 fibrosis. So this is the question we want to address with our biomarker discovery program. So what we have been able to identify is an algorithm, which is a score, okay, and what we have been able to find is if a patient comes in, we put that through the algorithm. If they're above a certain score, they are NAS4 or more, F2, F3 fibrosis, they should be treated. If they're below that score, they do not need to be treated. Okay, this is what we have in hand today. This is just a quick look at, if you look at the AROC, and here we're just comparing the performance of our two algorithms. So using our approach, I mean, we've measured over 80 different parameters using two different approaches, and at the end, using these two different approaches, they both identify the same 5 variables, okay, which we use on our algorithms. Right now, we have 2 different algorithms. And you can see here the performance. And to the question of identification of patients for treatment, clearly our 2 algorithms perform much better than any other scores, significantly better than the other scores which exist today. So here are the 5 parameters that are on the lower right-hand corner. Okay, you all recognize most of them. Okay. HbA, remember, Nash is the liver manifestation of metabolic disease. Not surprising to see HbA1c as an important component. The other one is the procollagen type 3. A2M is alpha macroglobulin. As some of you may know, it's a marker of liver disease as well as liver fibrosis. And then the 2 other variables are very important. We think this is a major innovation which we bring to the field, is to measure miRNAs. As some of you may know, miRNAs are related or involved in the progression of various different chronic diseases, cancer, metabolic disease, and so on. So these 2 mRNAs, MIR34A, 200A, has already been demonstrated to be involved in liver disease, liver fibrosis, as well as metabolic aspects of NASH and FLD. Okay, so these are the 5 parameters in our 2 algorithms. We have an abstract that's been accepted for presentation at EASL. It will be presented by Irene Sanyao, as many of you know, is one of the leaders in the NASH field. So I invite you all to come out and look at that poster. We will be providing quite a bit more detail about that program. So here's a quick overview of where we are and where we want to be going, plan to be going. So right now, I mean, we do have this score in hand. We will be further validating this score in collaboration with our different experts. Okay, they have their own cohorts, so we will be further verifying these 2 algorithms. It's also important to note that we will be using the samples of our phase 3 program to finally verify this algorithm. It's important that you also note that we will be doing this using the first phase of recruitment. So if we talked about recruiting 1,000 patients for the first endpoint, once we have those patients recruited, we will have their blood samples, we will have their biopsies and so on. So we will be able to already verify our score at that stage. We don't have to wait until the end of the trial. Moving forward, it would be important to be able to develop a kit to be measured these different variables. I think GENFIT, we're very good at analyzing samples and clinical development and so on. We are thinking maybe the best way to move forward is to partner with a company to develop the kits and so on. So these are things that we're thinking about as we move forward. In terms of timing, we're looking at the ability to be able to have this on the market, to make it available to patients and the physicians at about the same time when elafibranor will be on the market. So with that, I think we'll move on, and Sophie will talk a bit about our elafibranor program in PBC.
All right, so PBC, so primary biliary cholangitis, sorry. So this is, as some of you might already know, but it's a cholestatic chronic autoimmune disease that affects the bile ducts inside the liver. So it's the intrahepatic bile ducts that are affected. You see on the left side of the graph here of the image, whether bile ducts, because of inflammation, scarring tissue, are really affected, and you can see that damage. It's importantly, the diagnosis is through, you have 3 types of diagnosis, and usually have to have 2 of those 3 criteria to have the disease. So the main factor that is, it's elevated phosphatase, alkaline phosphatase. You also have the presence of antibodies, and these are the AMA, the anti-mitochondrial antibodies, and you have liver histology again that shows the lesions that are specific to PBC. The prevalence of the disease, it's a rare disease. It's only up to 0.05% of patients who have PBC. Importantly, you do have a patient profile. Around 80% of the patients are women that are around 40 to 60 years old. You have, for a long time, PBC can also be asymptomatic, but when it does become symptomatic, it's mostly through fatigue, very high fatigue, and also pruritus. So those are the main symptoms of the disease for the patients. And like I said, it starts off as being asymptomatic before the symptoms come in. And I would say that, I think, in the next slide here. So the pruritus is very important in these patients. It is one of the major symptoms, and that is very invalidating to the patients. You also have abnormalities that you can find in different clinical exams and blood sampling. So you do have increased gamma GT and immunoglobulin. You have the hyperlipidemia actually in those patients is, you have to understand, it's a high HDL, meaning it's the elevated good cholesterol. So they do have a cardioprotective profile with the HDL that is raised in these patients. So you want to keep that cardioprotective in these patients. They have also vitamin deficiency, osteoporosis. Those are secondary elements of the clinical disease that you can find in PBC. But globally, the specific symptom is pruritus. The fatigue, like I said, it's very important. You do have 78% of patients who have fatigue, so it's important for the quality of life of these patients. Pruritus and fatigue really make it difficult for them on a day-to-day basis to have, a quality of life that can be, the disease can become invalidating because of that. You do have association with other autoimmune disease in these patients. And very importantly, this is a very serious disease that leads to cirrhosis. It used to be called, the PBC used to be C for cirrhosis, now it's cholangitis, because all these patients, when the treatment does not work, and today there's only one approved treatment, which is UDCA, they just invariably progress to cirrhosis and to liver failure, and they die. It's really a very severe disease. Today, like I said, we have the treatment with UDCA. Importantly, the unmet need is still very, very present because you have Among the patients that are treated with UDCA, you have about 50% only of those patients who are actually well balanced with UDCA. The others can either be non-responders, they can be partial responders, or they can be intolerant also, to the drug, or have other pruritus also with these drugs. So when we look at where elafibranor can come in, and I'll explain why we believe that there's a very strong rationale for elafibranor to address this unmet need in PBC, the target population remains, globally on the partial responders, the non-responders, and the intolerant patients to effective treatments. I will not go into detail in this slide, but this is important to us because we have clinical data, which I will show with elafibranor, that is very interesting for the PBC field. But there's also the mechanism of action. As you know, elafibranor is a PPAR alpha and delta agonist, and the PPAR alphas, there's been extensive work. You see here the number of studies evaluating the PPAR alpha, the fibrates in PBC, and there are different mechanism of actions who actually help the transport of the bile acids out of the hepatocytes, and that also detoxify the bile acid and invalidates, the bile acids. Therefore, you have less damage. You prevent the damage to the cholangiocytes, provoking the damage in the bile ducts. So this is well known. This has been evaluated in several studies. And then we can look at also what we had in our clinical trials with elafibranor. And like we showed before on the slide through Manal in the phase 2 results, we do have a significant and important reduction in alk phos, and you see that in that chart. We had visits every 2 months, and you see the two lines on the bottom are 80 and 120 milligrams, so the decrease in ALP is very quick, and it remains low throughout the treatment, and you see that placebo does not move, or rather even increases a little bit on alkaline phosphatase. And this is according to the different quartiles, meaning the levels, initial levels of baseline for alk phos, where you see that the efficacy is always there with the doses of 80 and 120 milligram compared to placebo. So this is taken from the data from our phase 2 trial. So when we look at this, and this is why we have thought this through, and we are starting the PBC program here, we have the clinical evidence of our own data, but we also have the fact that this unmet need still remains, very serious condition, and we do not have adequate treatment today to treat the PBC. We have the ALP reduction that was proven in our trials, not only the phase 2 in Nash, we also had previous phase 2A program in different metabolic populations, and we always consistently had this significant decrease in alkaline phosphatase. We have the PPAR alpha activity on bile acids that is well known and demonstrated. We also have no pruritus with elafibranor, which is very important in these patients where the major symptom is already pruritus. And there was some trials also that have shown with the pan-PPAR a decrease in pruritus in PBC patients. So this is why for us, we're launching this PBC program with elafibranor. And to give you an idea on the timelines, we are launching this, the first pilot study, the phase 2 study this year in 2016. And it will be followed with a longer phase 2-3 trial once we get those first results. Of course, the regulatory input is very important. We've done this with our Nash development, always discussing, in parallel with the agencies, and we're working in the same way with experts and authorities on the PBC field, of course. So we will have this input. As you know, there will be input from regulatory bodies very soon on surrogate endpoints and approvals in the PBC for new drugs. So this will be important for us to continue working in the same way that we've always done with GENFIT, talking to authorities as a parallel process of our development plan. The other studies still with elafibranor, we do have, and I think we've spoken of that already quite a few times, but we are launching this year as well the first pediatric trial. So we will start with the PK/PD, meaning a pharmacokinetic and pharmacodynamic study, more on metabolic aspect, on decrease of ALT, but also on metabolic activity in a pediatric population. So this will be from 8 to 17 years old. And we will launch this year as well in 2016, around June or July for this study. And this will be followed by a long-term study once we get all this data on PK/PD, which will be more, the long-term biopsy trials. And this is, again, As I said, we're discussing this, of course, with the top experts in pediatric NASH, also through the Liver Forum. As you know, there's also a working group specific to pediatrics where we're participating as well to make sure that we follow through with the correct endpoints in development in pediatrics. And importantly, in children, the metabolic facet of the disease is also very important. Most of them are pre-diabetics, and that is very important to have that metabolic treatment as well as the histological treatments with elafibranor in this population. And for chronic treatment, you know that children, they will start the treatment very early. So of course, the safety and tolerability is also a very important factor for children, even more so in this pediatric population. The other study that we're starting now is the, so it's cirrhosis, but of course NASH-induced cirrhosis trial, and we will launch a pilot study because we will be looking at the first effect on HVPG, which is currently the surrogate marker for the cirrhosis trials. So this pilot cirrhosis trial on a, not a very large population, but to get the first information on HVPG before progressing to a phase 3 cirrhotic trial, and this will be done the second half of this year for the pilot cirrhotic trial. The combination therapy, so we're not, you're going to talk about the combo therapy, Dean, program?
There's 2 slides.
Yeah, very short, thanks.
So just to come back to NASH for a few minutes, this is something that we are looking into for quite a while now, and I think this is going to be something that's very important for the whole NASH field and for the whole population of NASH patients. And this also comes back to the objective of GENFIT to really consider the whole global clinical management of the NASH patients. On the left-hand side, I already talked about the fact that the compound elafibranor works both on histology as well as the cardiometabolic aspects. I talked about another important aspect for the patients is, of course, a non-invasive biomarker to identify patients for treatment. But the other important aspect to really, what's the best way to consider the whole population of NASH patients? I think we all know that the NASH population of patients is somewhat heterogeneous, and this is not at all surprising. Again, I repeat, NASH is the liver manifestation of metabolic disease, and we know metabolic disease by and large are multifactorial, different mechanisms involved, and so on. So what's clear is that if you really want to consider and treat the largest population of NASH patients possible, you should be considering combination therapies. Okay, and this is what we have been doing. What's important is that when you think about combinations on paper, it's not that difficult to think about being able to combine 2 different compounds. You think about complementary mechanisms of action, hoping that the effect at the end will be additive and even better synergistic. Again, that would be on paper. I think the most important aspect is you have to test these both in vivo models and ultimately in the clinic, of course. And this is what we have been doing, both using compounds which we have in-house, proprietary compounds of GENFIT, but also importantly, we are discussing with different groups, testing compounds which are in the NASH pipeline with the other companies which are also working on NASH. Here I'm just showing the progression of NASH from early stage disease to late stage disease. I think it's also important to note that if you consider elafibranor, which is able to treat quite a wide spectrum of patients, fairly early patients, because the compound does work on histology with that cardiometabolic benefit that we see, that's also going to be beneficial for early stage patients as well as late stage patients. But having said that, I think when you think about combinations, we should be thinking about the whole spectrum and where these different target genes or therapeutic targets lie. They could be early in the process, they could be later in the process, and I think we have to consider the whole spectrum of these different possibilities, and this is what we are doing. I think it's also important to note today, elafibranor is well aligned to be first-line treatment. When I say first-line treatment, when you think about the different programs that are in this space. I think that the clear differentiation of elafibranor beyond the efficacy on the histology is the other important aspect, of course, is the cardiometabolic benefit and also the tolerability, no side effects, no tolerability issues with the compound. Okay, so this, I think, clearly puts us in a position of first-line therapy, and because of that position, I think we can take the lead and think seriously and you know, be proactive and consider what we should be considered combining elafibranor with. So having said that, like I said before, this is a program considering both in-house proprietary compounds as well as with compounds that's in a competitive landscape. We're talking to different groups, and this is what we are doing in that space. Now with that, what I want to do now is just move on and talk about some of the other programs which we have ongoing at GENFIT. The first one I'll talk about is our program in fibrosis. Okay, I say fibrosis, our focus is mainly on the gastroenterology space, liver fibrosis, and we all know the importance of addressing liver fibrosis. And when I say liver fibrosis, it's not only NASH-induced liver fibrosis, there are other things, you know, viral-induced fibrosis, alcohol abuse, autoimmune disorders leading to fibrosis, and so on. So in our program, I think the idea, and this is clear, that you want to go in there and treat the patients, okay, with well-established fibrosis. It could be somewhere in the middle before they hit cirrhosis. Ultimately, in that stage, you want to prevent them from progressing to cirrhosis, but you can also treat patients later on, cirrhotic patients, okay, and prevent decompensation, for example. Having said that, our focus, of course, is on liver fibrosis, but the programs that we are working on, the compounds which we have in hand, it's important to note that they can be considered for other disease areas of fibrosis. Things like lung fibrosis, other disorders where fibrosis is an important component. These are things to be considered. These are not our areas of strong expertise, okay, and so when we have these things in hand, we think it'd be interesting to arrive with a proof of concept, at least a preclinical proof of concept, and look to partner some of those programs. Another interesting aspect that we are considering is the area of fibrosis in Crohn's disease, in IBD. Okay, it's clear that in Crohn's disease right now it's addressed mainly with anti-inflammatories, but we know that although the anti-inflammatory compounds do have good efficacy, it is not today decreasing the number of intestinal resections. It's not decreasing the number of surgeries that's required in Crohn's patients. And so in our discussions with the KOLs, and I think this is quite innovative, is that there is the fibrosis component of these patients, and this could be the key really to prevent these patients from undergoing surgeries due to their Crohn's disease. I think this is an innovative part of our strategy to think outside the box in some of these disease areas, and here the antifibrotic compound can make a big difference. So in our fibrosis program, just to run through it very quickly, what we did was we developed a screening assay. Okay, it's easy to look, you know, here's represented in one slide, but this took quite a bit of time to optimize. Basically, the cells involved in fibrosis, we all know it's a stellate cell. They are quiescent, and then if they're induced with different cytokines like TGF-beta, for example, they then evolve, differentiate into myofibroblasts or activated stellate cells, and this is the cell type which secretes extracellular matrix and leading to fibrosis in the tissue. So we optimized the screening assay and the readout. It's somewhat a phenotypic screening assay, and the readout here is alpha SMA, which is a protein intimately part of the fibrotic pathway. So two parts of this program. One part we had considered, profiled, and screened proprietary NCEs, okay, compounds belonging to GENFIT, and then the other aspect was to go into the pharmacopoeia. Okay, and screen the pharmacopoeia using this assay, and really to identify compounds which can be repositioned to be used as an antifibrotic. So these are compounds which are originally not developed for fibrosis, and we were able to identify 7 different compounds which have impressive antifibrotic activities. just to give you an idea of the antifibrotic activity which we have been able to demonstrate with the assays that we have in hand. Basically, the dots there just represents a subset of the different compounds which we had screened, and you can see that there are quite a few, the darker ones coming from the pharmacopoeia, and you can see that they are able to inhibit the secretion of alpha SMA, that's the marker, with a good potency. Okay, the EC50, IC50, it's in the low double-digit nanomolars, okay, which is quite potent compounds, and we also have a few proprietary compounds in that space as well. Here is a result looking at the antifibrotic activity in a model of fibrosis. Without getting into any details here, you can see on the two graphs, looking at either collagen or the gene encoding collagen. Clearly, you can see the compound decreases the level of fibrosis. And then if you look at the histology, top panel control, middle panel, you can see the fibrosis induced. On the bottom, you can see the effect of the compound, which is clearly antifibrotic. Okay, so this is just to give you some top-line data as to what we have been able to demonstrate with the compounds we have in hand. So having said that, this is where we are with the two sub-programs in the Nash and in the fibrosis space. So in terms of the NCEs, okay, proprietary GENFIT compounds, we are ready to go into preclinical development as of today. This is something that Bob is working hard on, and then we're moving forward with that program. And the other program, which is the repositioning of the pharmacopoeia, we should be ready, and we will be ready to move into a phase 2 program, okay, because this is compound on the market. It has a strong dossier already. We have in vivo testing, which is in the process of being finalized, and we will be able to move into phase 2 towards the end of this year. And of course, a very important aspect of the repositioning strategy of compounds in the pharmacopoeia is the intellectual property. Of course, these are things that we consider very early in the process, and so this program also has an intellectual property component to it, where GENFIT has applied for patents for the protection of this particular compound in the space of fibrosis. With that, I'm going to move on and speak about another program which is very exciting for us, which is a program in the space of autoimmune diseases or chronic inflammation, and this is targeting a nuclear receptor. Okay, remember, GENFIT has a very strong expertise in nuclear receptors. Elafibranor targets 2 nuclear receptors, PPAR alpha and delta. The scientific expertise coming from our scientific co-founder next to Jean-François, Bart Stiles, is one of the pioneers in nuclear receptor space. So ROR gamma T is a nuclear receptor. Bob has been working on this very carefully, and so ROR gamma T targets the IL-17 pathway. As some of you may know, IL-17, a pro-inflammatory cytokine. There is clinical proof of concept. There's antibodies out there where if you decrease IL-17, you have an anti-inflammatory readout, and this has been demonstrated in the clinic and compounds on the market and so on. So this is one of the mechanisms of targeting ROR gamma T, because that directly leads to increased expression of IL-17. The secretion of IL-17, like I said, is anti-inflammatory. Pro-inflammatory. Another important aspect of targeting ROR gamma T is that it is involved in the lineage commitment of T cells, okay, between Th17 cells and Treg cells. So Treg cells, on the right-hand side here, Treg cells will just to be simple, is the anti-inflammatory phenotype, and on the left-hand side, Th17 are cells which are pro-inflammatory. And what ROR gamma T is able to do is to affect this balance between the Tregs and the Th17s. Okay, it can favor the Treg over the Th17, therefore favoring the anti-inflammatory population of these cells. Now, the indication that we are really addressing with this program, like Jean-François mentioned at the beginning, is an autoimmune disease, and it's autoimmune hepatitis. It is a progressive disease. Right now, there are they are being treated with anti-inflammatories, okay, steroids and so on. We all know steroids over long-term treatment do have side effects related to it. So there is this very large unmet need to be able to treat these patients in the long term. This is very long term, if not chronic treatment of these patients, okay? And so what we think that we should be doing is to be able to identify compounds which are inhibitors of RR gamma T to be able to keep these patients on treatment and in their repressed state in the long term. Tregs, we know, are important. And when you think about autoimmune hepatitis, it's important for the self-tolerance and maintaining that in the liver. Also, we know that in autoimmune hepatitis patients, the number and function of Treg cells come down, and also that they do lose their function in terms of the ability to be immunosuppressive, okay? And it's also clear that, I mean, I think it's one hypothesis, and this is going to be borne out based on our results, is that if you're able to inhibit RR gamma T in these cells, that you should be able to increase the number of Tregs as well as the function of Treg cells. So this is the reasoning moving forward. Just to give you a little bit of results here. Here we're looking at the T cell skewing assay, basically looking at the ratio between the Treg cells, again, okay, the anti-inflammatory cells versus the Th17, the pro-inflammatory cells. So basically, I won't get into the details here. When you look at one cell population, you hit it with the inhibitor, you look at IL-17, you can see this compound is able to repress it. And then on the bottom side in the green there, if you start with another subpopulations of cells, okay, and you hit it with the compound, there you're able to maintain the level of FOXP3, which is a marker of Treg cells. So basically, the take-home message here is that the compound, an inhibitor of RR gamma T, one of our compounds, is able to affect the ratio between the Tregs versus the Th17s, which is a very important point, of course. So this is where we are with this program today. We are moving forward with a lot of focus on this, and we will be ready to go into preclinical development, okay, in the second half of this year with this program. So with that, I think I'll stop. I'll hand it back to Jean-François to provide an overview.
Okay, before coming into Q&A session, I would like to give you some key messages and to summarize what we just have said during the last hour. First of all, Nash remains our primary focus, as you can imagine, and we consider that elafibranor as a first-line potential, and we have explained why we believe strongly in that hypothesis. The phase 3 Resolve It, we have already involved first patients, and as we have said, we think in the next 12-13 months we will have the first 1,000 patients that we need for the Subpart H and for NDA. And on the diagnosis field, we consider that our biomarker program, with the same deadline, I will say, with an approval around the approval of the drug, is the only way to unlock the market, and unlock the market not only for elafibranor, but for all the drugs in this field. You understand that today we are announced different things in the Nash field, and particularly we consider as very important what we are doing in the field of pediatric, because it's really a very big health burden, and particularly in this country, but not only. And we consider that we will do that in perfect collaboration with academic and regulatory agencies to be sure to address correctly this population, with no solution today as well for adults, but I think it's a particularly strong motivation for GENFIT to be involved in this pediatric area. I think Dean has described that combat therapies is something that everybody is thinking about, and we want to be proactive in this field. We do not want to have in some years just, I will say, people trying their own recipes to combine this drug with this other one and so on. We want to provide proof of concept of synergy between some drug with different and complementary mechanism of action. It's why we are strongly working on that. And of course, I think you understand that we will discuss with different companies to see what we could do together to be proactive in this direction. GENFIT is, as announced today, a new PBC program, but you have understood that we have done that after many months of thinking about and after clinical data and clinical evidence. We have discussed with a lot of KOL in the field, and it's a decision which is taken because of the PPAR alpha activity on the relevant PBC targets. And the second aspect is that we have seen in all the phase 2 that we have a real potential to reduce ILP, which is one of the main factor for getting an approval in the field of PBC. Of course, in PBC, we know that many things will happen in the following days, months, and we will pay a very big attention to what is the conclusion of the different meetings with the FDA and the EMA in this field, and we will, as Sophie has said, we will be very close to the regulatory agencies to progress in the PBC program. So we want to continue to target unmet needs. I have said at the beginning, we want that to be in coherence with what we know the best in GENFIT. I mean, metabolic disorders, metabolic disease, autoimmune disease, and now we consider that still many unmet needs in the gastroenterology field, and we want to be very present in this field, not only as an R&D company, but also as a commercial company. Now, it's important to speak about this agenda because, many people think that now phase 3 in Nash, we have to wait 2-3 years. No, I think it's not eternal. We will have a lot of things, a very strong news flow in 2016, in the following months, in 2017. And I think it will be done of course, the progression of the enrollment for Support H, and expectation of 1,000 patients in H1 2017, an expected NDA in 2019. For the biomarker NASH, we also will take the profit of the phase 3 to validate it and to accelerate the regulatory pathway to go for an approval approximately at the same time as a Support H for the drug. PBC, as we have said, we are starting the enrollment in H2 2016. We will launch a pediatric NASH in H2 2016 also, and the start of human testing for the lead compound that Dana just described a few minutes ago in liver fibrosis will also begin in 2016. And the oral gamma T will be in preclinical development at the same time. So we do that in parallel with assessing potential initiatives in licensing and in coherence with the pipeline we want to build. And all of these things, I will say, it will be my conclusion, as a result of many years of big efforts in the R&D field, but also the fact that we have decided a very clear strategy for years now, and that we are continuing to progress on this route. Of course, as you know, we are a very resilient company. We consider that even if there are some hurdles, it's not a problem, and we will pass over that. So thank you for that. I think we are going now into Q&A session. I don't know if you have some question from the room, or if we have some papers coming from the room also. So Dean, if you want to be the chairman of that.
I have all the fun.
For your biomarker program, do you expect additional validation will be required for the pediatric group in NASH? Or is there any crossover between the adult biomarker program and the pediatric program?
Okay. Just to repeat the questions for those who are listening in by WebEx, the question is for the biomarker program, whether we think that additional validation will be required for the pediatric population versus the adult NASH population, and considering that what we have done so far, of course, is from the phase 2B, which is the adults. I would think at this point that this is something that should be considered and looked at and before it can be used in pediatrics. So that's my thinking of this, but I think I can let Sophie chime in on that.
Sorry, just want to check. Maybe Manal, you can add to that. But it is a very important question, and I think it goes beyond the biomarker. Just the fact that pediatric NAFLD or NASH is different, even histologically. You know, it's not defined in the same way, so it seems logical that you're not going to, you know, address the same specifics with biomarkers as well, because you have to link it to biopsy as a first stage. So yeah, this is for me also a separate validation that needs to be done in the pediatric population.
Yeah, I agree completely. I think there are very unique histologic features in pediatric NASH that we do not see in adult NASH. And the mechanisms underlying pediatric NASH are somewhat different even physiologically, particularly with the alterations that may occur during adolescence, hormonal changes through puberty. And so I think it's going to be essential to have independent validation in pediatric NASH. We may come to learn that the biomarkers are comparable to those in adult NASH, but I think pending the further validation in independent cohorts, we won't know.
And then we looking at additional endpoints or different endpoints also in that case. And then in PBC, can you just go through the rationale for not looking at histological evidence or antimitochondrial antibodies? Because I don't think that was an endpoint in your PBC studies.
The question was for the PBC program, what are some of the other endpoints which should be considered?
Yep. Yeah, no, it just, and I think that's important again for the endpoints. And we're lucky to have Manal, who actually trained with Keith Linder in PBC and started also in cholestatic, so I'm sure she can add to that. Today, the question around PBC is, you know, the question around the endpoints. And like Jean-François was saying, we do need to have, you know, the first feedbacks from the authorities. We'll have a very soon, I think, following the advisory committee early April, on the trials with OCA, and I think that's very important for the field of PBC to know what the reaction will be from the FDA, the approvals, the surrogate markers. So we're just talking about what I mentioned here was the primary surrogate marker to start with the ALP. But of course, you know, the other endpoints that you mentioned, that's part of the disease and that's part of what will be looked at. But to start off with, for the, you know, the pilots, the proof of concept, just to make sure that we have all this, you know, needed decrease in ALP. But of course, a longer term PBC trial will include all these other endpoints.
I might add, especially with PBC, at this early phase of a treatment program, improvement in liver biochemistry, predominantly alkaline phosphatase, GGT, alterations in antimitochondrial antibody changes are enough to justify the proof of concept. Keep in mind that the FDA regulatory authorities, even in PBC, will pay particular attention to alterations in quality of life measures. So even for a phase 2 study, early phase study, if patients itch less, feel better, and certainly survive longer, which would be proven in subsequent studies, that in of itself is a significant measurable clinical outcome.
Okay. Last question. For your preclinical program, you had mentioned for your IP that you're going to get protection around use in treating fibrosis, but is there concerns with freedom to operate as far as other competitors having a more broad coverage and you interfering by having that use in this space?
Yeah, the question is for the program on fibrosis, where we are repositioning compounds in a pharmacopoeia for targeting diseases in fibrosis, whether the IP should also consider the FTO freedom to operate. Definitely, these are things that has to be considered. We consider this very early on in the process, and the strategy of GenFit surrounding this space is something. Of course, this is considered, and we will have freedom to operate on this particular compound in the area of fibrosis.
Just one word. When you are working on repositioning strategy, I will say 50% of the work is to work on the medical aspects, scientific aspects, proof of concept. But 50% of the work is to work on intellectual property. It's really a big part of this strategy. We have some questions on the papers. Coming back to Nash, or 2 questions on Nash.
Oh, here's one. Okay. Question is, for the Nash phase 3 program with elafibranor, why are we considering only patients with F2 and F3 fibrosis, and why not the F1 patients? I think that's a very good question. What I can chime in, and then I think I'll hand it off to Sophie and Dr. Abdelmalek, who know this much better than I do. What I can say for sure is that when you talk to patient advocacy groups, this is something that they really want. They want to be able to address NASH patients with F1 fibrosis with the understanding that they would rather have a treatment to address these patients before they advance onto later stage disease. So that's just my top line input on this, and I'll let Manel continue.
So I think we've come to learn that, at least as it pertains to liver-related morbidity and mortality, the primary predictor, and no other primary predictor, not necroinflammation, not steatosis, but the primary predictor for outcomes is fibrosis. So in keeping with the goals of the FDA and trying to alter the natural history of the disease, it would make sense to enrich a study cohort with patients most likely to develop disease progression. So we a priori are selecting those patients at high risk, which would allow us, in the capacity of a phase 3 study, to discern measurable outcomes in associated morbidity and mortality from a liver disease standpoint. By selecting out patients with stage 0 or even stage 1 disease, you are actually enriching the patient cohort for those less likely to develop those outcomes, because the natural history of the disease progression from one stage to another is on average about 7 to 10 years. So at this early stage, it makes sense to certainly enrich the cohort with patients who are going to develop such outcomes or at highest risk for such outcomes. And at the same time, as we learn more about this space, these therapies, we would certainly want to be able to treat all patients at risk and open up the space to phase fibrosis stage 1.
Metabolic conditions.
Just if I can add to that, in our study for the phase 3, and this was discussed as well with the authorities. So we are adding, an exploratory arm with the F1 patients, but importantly, the F1 patients who will be enrolled and recruited in our study and followed to the end are enriched with cardiometabolic risk factors and a NAS severity that is higher. So we have criteria for the F1 patients in our study that make them high progressors to evolve faster to the end stages of evolution of fibrosis.
Okay, thank you.
If there are no questions coming from, I got one here that's written down. Here's a good one, maybe for Jean-François. What about the pricing in elafibranor when you consider PBC and NASH?
Great question. As you know, the agenda we have for the NDA, I think we will get probably the approval, potentially the approval for the NASH, and the approval for PBC if we succeed in this field roughly at the same time. So I will say that we cannot envision to have a different price for PBC or for NASH. There is no reason for that. And I think it will be interesting for all the PBC patients which are more a rare disease, and usually the companies can get better price for that. But because we will have the two drugs approved at the same time, we will have the same price, and the same price will be probably the price for NASH. So it's not really for us a problem because of the agenda. Of course, I can imagine that it could be more a concern for some companies with a different agenda for the PBC approval and NASH approval, but it's not our case.
Here's a question coming back to the biomarkers. Why is GENFIT so confident that you'll be able to bring something new or better to the market? Good question. I think there are different aspects to that question. I think first of all, when we say we want a non-invasive biomarker to identify patients for treatment, I think that in itself is something that's different than what other groups have been doing. I know some other groups, many groups are trying to find biomarkers to represent the whole spectrum of NAFLD to NASH patients, which is not an easy thing to do. I think the relevant question from a clinical standpoint is to have a biomarker to identify the patient for treatment, and this is what we were presenting here today, that we are addressing, at least for now, okay, to have a biomarker to identify patients with NAS4 and more with F2, F3 fibrosis. So I think that's an important distinction. I think another innovation which we bring to this field is that we are measuring an important parameter or type of parameters, which is the mRNA. I mentioned briefly that mRNAs are involved in the disease process in many different chronic diseases, and this has also been demonstrated for NAFLD and NASH. And so this, I think, is important when we think about non-invasive biomarkers. And we all know that, mRNAs are released into the circulation when the different tissues are stressed, and this is also could be part of the pathophysiological process. I think it's also important that as we think and move forward, just to stay on the theme of mRNA for a minute, that at the moment we are still looking at additional mRNAs. Okay, they are not all described, not all of them have been described to be involved in liver diseases either. So we do have the sample sets in hand where when we compare those patients who should be treated versus those who should not be treated, we know there's a whole series of mRNAs which are upregulated or downregulated. I think these represents additional mRNAs which could be imputed into the algorithm to improve even further the algorithm scores. Okay, so I think these are different aspects why we think that we'll be able to be successful. And before I end on that, I think the sample set that we have is also very important. We do have the sample set from the phase 2b trial. Okay, and this is a rare sample set, samples coming from an interventional trial. It's a double biopsy controlled trial. And like I said, samples collected throughout the process, a 1-year treatment process, where a significant portion of patients evolved from NASH to non-NASH. I know this is an invaluable sample set because we do have various different groups who's been contacting us over the last few months, many months, trying to access those samples, okay, to help them verify what they have in hand in terms of biomarkers. So I think right now the strategy, of course, is to use these samples for our own biomarker discovery, and if we need to partner and provide those samples, we will be looking into that as well.
Can I chime in with a comment? I think we sometimes lose sight of the fact that NASH is a very heterogeneous disease. And if I were to envision where we will be in, hopefully in another 5 to 10 years from now, we're not going to have one biomarker. One size will not fit all. And we will not have one therapy. We'll have a landscape of treatment options. And I think the biomarker discovery for GENFIT will be uniquely poised, potentially, One, of course, to discern those with highest risk for disease progression, discern those with NASH and fibrosis, but also to lend predictors for cardiometabolic risk. There are patients who walk into my clinic who may be 500 pounds who have no lick of liver disease, and others that walk in moderately obese whose primary cause of death may be cardiovascular as opposed to cirrhosis progression. I have patients who progress to cirrhosis who never have heart disease, and others who die of their heart attack and have little or no fibrosis. So ideally, as we put therapies into practice, discerning who's at risk for what, personalizing the treatment, identifying who's at risk, and potentially predicting responders from non-responders in any cohort treatment is going to be ideal. And so I think to implement biomarker discovery in the context of these studies is visionary and actually plans ahead for defining the most ideal populations to treat with that unique therapy in that unique space to decrease a unique patient's overall risk for morbidity and mortality with whatever inflammatory, cardiometabolic, and or liver-related markers may be in whatever panel is defined for that cohort.
Thank you. I have here a related question. I think Dr. Abnomali can maybe further address this. I know you already addressed it during your presentation. The question is, what more can we say about the metabolic conditions of NASH patients?
So clearly the metabolic conditions of NASH patients characterized by truncal obesity, the apple paradigm as opposed to the pear. So adiposity distribution centrally. With that comes increased risk for hypertension, diabetes, high panel of total cholesterol and LDL cholesterol, low HDL phenomena. So what has been the challenge for metabolic syndrome in general, you will also see. Patients, as they progress in years, will have higher risk for fibrosis progression. So we will typically see fibrosis in patients who are older than 45 to 50 years of age, with the highest risk and predictor of fibrosis, at least clinical, being the presence of well-established type 2 diabetes, which may carry anywhere from a 6 to 13-fold increased risk for fibrosis.
So thank you very much. I think we have two last questions around PBC, and then we will say hello to everybody. Okay.
Yeah, I got two questions here on PBC. I think I'll combine them. Bob, you can already start thinking about it as I read them out. One question is, in the space of PBC, when you think about PPAR alpha, which is definitely a good target to address for the treatment of PBC, how would you position that versus the fibrates, okay, which today can be used off-label? And then a related question, in PBC, what differentiations are you expecting with elafibranor versus other drugs? Okay, so I think for the first question, yes, there are fibrates out there which have demonstrated efficacy in different models, even in the clinic. I think definitely when you think about the treatment paradigm, something that is agency approved versus something that's not, something that's prescribed on label versus off label, I think that does have a major impact. I think that's the easy part of the answer. There's something else I want to raise. When you think about elafibranor, we spoke about this in the past, the elafibranor undergoes enterohepatic cycling. Okay, the fact that it does undergo hepatic cycling, what we do know is that there is a concentrated, a higher level of the compound found in the bile ducts. Okay, so when you think about PBC, of course, bile ducts having a compound which targets PPAR alpha and PPAR delta, which has a higher concentration in the enterohepatic cycle in the bile duct, I think this is something that's important, and we can think about having increased efficacy with a compound like that. differentiation between allofibranor and other compounds.
So it's important to consider that elafibranor is a PPR alpha and PPR delta agonist. So you all know that PPR alpha is highly involved in bile acid metabolism, so that's not different from the fibrates, other fibrates. The differentiating part is probably the PPR delta activity, which is highly involved in the management of inflammatory disease. And we know that PBC has a high anti-inflammatory component. And since our drug is actually nicely exposed both in the liver and in the bile system, we think that this is probably going to be the major difference as opposed to the fibrates.
Okay, thank you, Bob. We have received by mail a question from a European analyst, so Dean, you can take it.
Yeah, so I think this is a question addressed to Dr. Abdelmalek and Sophie Meunier as well. So the question is, on slide 13, we refer to an updated definition or resolution of Nash. Basically, it focuses on ballooning and inflammation. If we can say a few words about where did that come from, how did we arrive at that?
So where we came from, our initial understanding, 2 decades ago, we thought fat was completely benign. We came full circle to think that the presence of fat was detrimental, only to learn more in the space that the fat really had no prediction for progression of fibrosis. The presence of necroinflammation and ballooned hepatocytes were the predictors not only for fibrosis progression, but for increased risk of cardiometabolic risk. Fat, we also learned, can flux in and out of the liver very quickly and very readily. So if one consumes an abundant amount of calories, fast food, you can get transient fluxes of fat in and out of the liver, and that storing triglycerides is actually a safe way to store fat anywhere in the body. When you develop a stress response, whether it's inflammatory, immune, in the presence of fat, you develop necroinflammation, ballooned hepatocytes, and that presence of necroinflammation, ballooned hepatocytes, is the primary predictor for fibrosis. So we moved away from the philosophy that fat was important, but that the injury subsequent to the fat, in some patients and not all patients, was the primary predictor of outcomes. And that's been shown over and over again, that the fat in and of itself can flux. The fat in of itself can be stored safely. Isolated steatosis without any measure of injury carries no increased risk for morbidity and mortality. And so we moved away from the philosophy of worrying about the fat and focusing more on injury.
Maybe, Sophie, you can chime in with this endpoint acceptability by the agencies.
Right.
And just, Sophie's also a formal part of the Liver Forum. She's co-chairing one of the working groups on disease definition and endpoints and so on. So I think the Liver Forum and the Nash space is a very important organization. So maybe you can say a few words, how the agencies as well as Liver Forum is implicated in this updated definition.
Yeah, well, as Manal was saying, and she's part of that subgroup as well, and we've been working on this. So, I mean, the Liver Forum, the construction of the endpoints for clinical trials, and this look at this new definition, all this has been, moving forward like for the past 2 years, with a lot of exchanges in the Liver Forum, with the presence of the FDA and the EMEA, with the ASLD, EASL, all the academic persons, but also industry stakeholders as well. So all these discussions have been done, in a consensual way, with working groups working, like Manal was saying, what is isolated steatosis, is it linked or not to outcomes? What is, the ballooning? Is it linked or not to outcomes? What is inflammation? And then what is NASH and what is resolution of NASH? So all this has been really discussed with all the major stakeholders through the Liver Forum, and not only the meetings, but external discussions and working groups and web conference that we've been having. And plus, there's a collection of data that's coming out as we move forward, and with the relationship to liver outcomes. Remember, the important long-term clinical benefit is what counts for all the regulatory bodies. And as Manal was saying, for the endpoint of the phase 2 trial that was modified with this definition, this came as a request. It's not just that it was accepted by the authorities, it came as a request from the authorities to go back to our phase 2 data, to look at this new, more stringent modified definition, and to apply it for the phase 3. So it's really something that we've been doing together all along, and it was a request from the authorities, and then a line, of course, to be used for the phase 3 and for most of the new trials coming on board that are taking ballooning as the major driver of disease, so the one that we want to have disappear to treat the underlying NASH disease.
Okay, thank you. We have one last question that came in, and I think it's an answer that only a CEO can answer, so I'll let Jean-François ask it and provide the answer.
And it is the last question. What is the question? No, he just told me that somebody was raising a question about financing the pipeline. I think it's a perfect good question. I will say that we have raised recently money, so as you know, we have about $130 million in bank. That means that we have money for the next 2 years. But of course, we do not consider that it's sufficient to develop all the company in the next 3-4 years. Do we expect to raise more money? Yes, why not? But it's not a question of today, We plan to be on a NASDAQ, but the question is only when the market condition will be better. So we are not in a hurry for that. We are just expecting good conditions. But you remember that we have repeated very often that we are discussing with pharma companies. We want to be present of the market, but not alone, of course. And we are thinking about co-development. We are thinking about co-commercialization, retaining some rights in some territories, but that means that we need partners, and we need partners to prepare the commercialization, and not the week of the NDA just to think about a partner. So I confirm we have ongoing discussion with partners, with different partners, about the GFT 405, elafibranor, in NASH, of course. So that means that we will probably get more money from these alliances to continue to finance the development of GENFIT and to continue to progress in the strategy we have decided and we have presented this morning. So it's the best conclusion for me is to thank you again for attending in this room. Thank you again to the audience for the WebEx session. And you will have a slide deck. I don't know if it's 10 minutes or in 2 hours, but you will have the slide deck we have presented here with some added slides giving more details in the day today. Okay, so thank you very much.